Hi, my name is Dave Gordon. I'm a bioinformatics software developer at the Institute for Genomic Medicine at Nationwide Children's Hospital. Today, I'd like to talk a little bit about how we're using machine learning to help classify variants of uncertain significance and guide the care of patients with lamin AC cardiac disease. First off, let's ask the question of what is lamin AC? Well, it's a group of proteins encoded by alternative splicing in the lamin A gene, which consists of an N-terminal head, a helical rod, and a C-terminal tail. Variation in lamin A can cause a spectrum of diseases termed laminopathies, but variants, especially in the rod and head, are associated with cardiac phenotypes such as arrhythmia and familial dilated cardiomyopathy, denoted cardiac laminopathies. Now, dilated cardiomyopathy due to this variation in lamin A has a poor prognosis, and this and other phenotypes often require surgical intervention with pacemakers or implantable devices. A patient was referred to us with a history of arrhythmia and atrioventricular block, that is to say, the electrical signals could not travel from the atria to the ventricles in their heart. They came from a family with a 4-generation history of sudden death and arrhythmia that showed a likely autosomal dominant inheritance model. The patient had been sequenced by a commercial lab, which returned a variant, D136E, in LMNA as a variant of unknown significance. This means that not much clinical action could be taken, and so we took this on as a research project. We Sanger sequenced 7 members of the family, 4 affected and 3 unaffected, and found that the variant segregated with the disease exactly. We also did whole exome sequencing to rule out any other genes or variants that might be causative. We found in there that the variant still segregated with 3 affected and 1 unaffected member, and didn't see any other variants that could be potentially causative. We classified this variant as likely pathogenic per ISMB guidelines given the additional information that we were able to obtain, and functional tests showed lower protein and mRNA expression in cell lines from affected subjects than those from unaffected subjects. So this raised the question, how could we translate this existing knowledge and computational predictions on variants in LMNA to improve medical care in cases where this type of family sequencing is not feasible? And so we developed a hypothesis that through machine learning, we could determine novel LMNA variants' similarity to existing variants in the base of knowledge and augment clinical care by translating these computational predictions into a better informed medical care and improve our patients' outcomes. Initially, we intended to train a machine learning classifier based on the existing knowledge of benign and pathogenic variants in gnomAD and ClinVar. However, this proved impossible as only one variant passing our filtering methods was labeled benign in ClinVar. So lacking the ability to train a classifier, we decided to use clustering in an attempt to segregate LMNA rod and head missense variants into groups. We used UMAP projection over all known LMNA missense variation in gnomAD and ClinVar using computational predictions from REVEL, DAN, MetaSVM, and polyphen2HDiv as our features. After projection, we used k-means clustering using the elbow method for cluster count and grouped the variants into 3 clusters. The results of our experiment supported our hypothesis. We generated 3 clusters of variants which showed significantly different rates of pathogenic variance between each. Clusters 1, 2, and 3 had percentages of 38%, 15%, and 6% pathogenicity in ClinVar, respectively. Our D136E variant from our family in our research study clustered with cluster 2. We also noticed that there was a significant difference in the number of pathogenic rod and head variants versus other pathogenic variants in cluster 2, which may merit further study. I'd also like to highlight some possible future directions of our work. While we're not quite to the point where we can directly drive care of patients with variation in LMNA, we believe that our clustering and variant risk stratification can begin to augment and inform our clinical decisions, especially in cases where we can't sequence the entire family due to consent or availability of samples. We speculate that taking variants from clusters 1 and 2 and doing further functional analysis would yield a high rate of positive results, and this would further feedback to strengthen the model in the future. We'd also like to add further biological and computational features to our machine learning model, which might help increase our ability to cluster and reveal further insights into how LMNA functions. And finally, we'd like to take this approach and apply it to other genes and genomic regions with limited known benign variation, with the hypothesis that this would also be applicable in those cases. Thank you.